Now I would like to introduce the speaker of today, Ulrike Topf. Ulrike Topf graduated from the University of Freiburg in Germany. She did her doctoral work at Friedrich Miescher Institute for Biomedical Research in Switzerland and obtained her PhD in biological sciences in 2011. She completed her postdoctoral training in International Institute in Warsaw and in CeNT of Warsaw University. And since 2018, she's leading an independent research group at Institute of Biochemistry and Biophysics, Polish Academy of Sciences in Warsaw. Ulrike's research interests focus on the identification of cellular mechanisms to maintain protein homeostasis during stress conditions and aging. She uses yeast and nematode C. elegans in her research as model organisms. Her research work contributed important findings on post-transcriptional response mechanism upon mitochondrial stress. This included modulation of global protein synthesis and protein degradation. Ulrike received scholarships from Swiss National Science Foundation, from Polish Ministry of Science, and L Oreal Poland for Women in Science. And her research is funded by National Science Center in Poland. So let's start. I would like to ask Ulrike to switch on microphone, camera, and good afternoon, Ulrike. Many thanks for accepting our invitation. And you can share your presentation and we can start your lecture. Okay, right. I think it's perfect. It's perfect. You can start. Digital floor is yours, please.
Okay, good afternoon, everyone. I thank, first of all, Adam and the Polish Biochemical Society for giving me this opportunity to present the lecture today. And as Adam already introduced, in the recent years, I was focused on studying mitochondria and how mitochondria actually signal their needs. And this will be the main topic of today's lecture. And I hope to give a nice overview of the current field in mitochondrial signaling during health and also during stress conditions. So mitochondria will be the central player of this talk. And actually, from the evolutionary point of view, mitochondria are really successful. So they derived when chemo and phototropic bacteria were taken up by archaea, and it developed two organelles. And since then, they had quite a career, and it's always viewed that mitochondria, of course, are the most important thing inside a cell, and they fulfill plenty of tasks. To get everyone on the same page, here you see a mitochondria. It's a beautiful organelle. It has two membranes, an outer membrane and an inner membrane, and it forms several compartments, the intermembrane space and the matrix part. And of course, mitochondria are most famous for the production of ATP, but they fulfill plenty of other functions as well that make them actually essential. And this includes signal transduction as well as calcium homeostasis, biosynthesis, bioenergetic metabolism, but also they influence the redox state in the cell and are important during innate immunity and more recently are they involved in aging-related responses, and of course, programmed cell death. Because they fulfill so many functions, it's easy to understand that if mitochondrial function fail, this is detrimental for the organism, and many human diseases are linked to mitochondrial dysfunction. But first of all, in a healthy organism, there are healthy mitochondria, and they're responsible for the growth and adaptation, energy, and they also produce heat. When something goes wrong in the mitochondria, do not switch immediately to a dysfunctional state. It's not a black and white picture, but rather they start to send signals to signal that something is wrong with the organelle and it needs to be attended to. And these kind of signals include, for example, changes in calcium homeostasis, or the increased production of reactive oxygen species. And such changes, they induce signaling coming from the mitochondria. However, if mitochondrial function cannot be restored, then mitochondria themselves become a problem and they lead to the diseases. So if we have problems with mitochondrial DNA, too much ROS production, we start to have inflammation, or this can even lead to the death of the cell. Now, we cannot just, because this important function of the mitochondria are involved or linked to many human diseases. And these are not just mitochondrial diseases, where the mitochondria themselves are dysfunctional, but there are many other disorders that are linked to mitochondrial dysfunction. However, there are so plenty of them, and not for all of them it's known whether mitochondrial dysfunction is a cause or a consequence of the disease. Now, these days, a big focus lies on the neurodegenerative diseases, but also on aging, which is of course not a disease by itself, but many aging related diseases are also linked to mitochondrial dysfunction. Now, we cannot just view mitochondria just as the organelle that fulfills all these functions, because of the evolution, mitochondria function very much depends on the cell that surrounds it. So despite that mitochondria have their own genome, only 1% of mitochondrial genes are encoded in the mitochondrial genome. Most of them, the majority of them, are encoded in the nucleus. They need to be transcribed, and mRNA is translated on cytosolic ribosomes. The nascent polypeptides are formed, and the proteins are kept in an unfolded state due to chaperone action and are then actively transported into the mitochondria. And their proteins fold and are distributed into the different compartments. So just because of this view, mitochondria depend on nuclear encoded genes, it is easy to understand that mitochondria need to signal to the nucleus. This kind of signal, called retrograde signaling, was discovered about already 30 years ago. So retrograde signaling is generally a transcriptional response. And it was discovered because of mitochondria that had compromised respiration. a signal is sent and signal cascade is induced to change gene transcription in the nucleus. Then genes are produced and proteins produced that help metabolic reprogramming and to restore mitochondrial proteostasis. This kind of signaling was first discovered originally in yeast, Saccharomyces cerevisiae, and was named retrograde signaling. It was about 1998, coming from the Butov lab. In the consecutive years, such signaling, such mitochondrial nuclear signaling, was also discovered in mammalian cells, although not exactly the same way than it was in yeast, because the proteins that are involved in yeast in the signaling are not conserved in mammalian yeast. So their so-called retrograde signaling goes via calcium signaling and NF-kappa B activation. And about the similar time, it was recognized that not only compromised respiration, but also problems with protein quality control inside the mitochondria can induce a transcriptional reprogramming in the cell. And this was originally discovered in mammalian cells, and it's termed mitochondrial unfolded protein response. And we now know that this mitochondrial unfolded protein response in the mammalian cells goes hand in hand with the integrated stress response regulating translation. However, mechanistic findings have been done during many, many years, mostly in the model organism C. elegans. And they're using genetic tools. The mitochondrial unfolded protein response was described in more detail. So this development took many years, but now just have a look what happened in the last 5 years of research. The picture looks about like this. And while there are still transcriptional responses discovered, there are coming more and more responses that are post-transcriptional and also post-translational. And it's not important to now describe you every single pathway that was discovered here, but have certain things in common. First common thing is that it's usually a response that is induced by blocking the import channels into the mitochondria, or by accumulation of the proteins that are produced in the cytosol but cannot enter the mitochondria. So the development was really vast and also very quick. And I show you here in the green boxes that the researchers that put a lot of effort inside, they also tried to compete themselves with finding most attractive name for the pathway that they developed. However, I think the future, it will turn out that many of these pathways that are now described separately are to some extent linked or even overlapping. So what do they have in common, these pathways? They have in common that they regulate the protein proteostasis network. So most of them, if not all of them, influence to some extent the free post of proteostasis, protein synthesis, protein folding, and protein clearance. So these are true post-transcriptional processes. And similar to the transcriptional processes, they aim in restoring a protein homeostasis, but not only in the mitochondria, but also in the entire cell. And they are activated to protect the cell from cell death, give metabolic adaptation, and as I said, to maintain cellular homeostasis. Today, I want to show you in detail, a little bit in detail, how we found that stressed mitochondria activate the proteasome and decrease protein translation. This pathway was termed APRAM, and the work has been done during my postdoctoral time in the laboratory of Agnieszka Hacińska, together with a really great team in her lab. So what did we study at this time? We had mitochondria, and we asked the question, what happens if we block import into the mitochondria? The research has been done in the yeast as Saccharomyces cerevisiae, and we used mutants of the mitochondrial import pathways. Now, proteins of the translocases are essential in the yeast, and so you cannot just delete them, but you have to use conditional mutants. And you see this here on this little picture. When you grow wild type yeast, they grow on permissive and restrictive temperature. But if you have a mutant cell that has a compromised mitochondrial protein import component, then cells only grow on permissive temperature, but their growth is, they are not growing on the restrictive temperature. So we took an unbiased approach and asked how the proteome changes if we block protein import. And Lydia Frube performed a SciLac approach together with the laboratory of Bettina Warscheid in Germany. And to our surprise, we found that it was non-mitochondrial proteins that were increased in the mutant compared to the wild type. Here is depicted two proteins of the that make proteasome assembly. So these are two chaperones that are necessary to assemble the protein degradation machinery in the cytosol. So having this initial results, we thought a logical conclusion from this was to check, so how functional is actually the proteasome in the import mutant. And we measured the proteasome activity. And the result you can see here, we found in the mutant, proteasome activity is significantly increased. And this could be partially reversed by expressing the import component from a plasmid. At the same time, we had proteasome activity increased, and we looked at ubiquitinated protein species, and with growing time at restricted temperature, we saw that increased proteasome activity had an effect in digesting more of ubiquitinated proteins. So this was a great finding, but what was the signal for this? We actually thought to test whether the proteins, the pre-proteins that cannot be imported into the mitochondria and are those mistargeted proteins and stay in the cytosol, whether these ones could mediate the increase in proteasome activity. So we overexpressed mitochondrial proteins because we knew that some of them at least will accumulate in the cytosol without having any mitochondrial dysfunction. Yes, it was possible to uncouple mitochondrial dysfunction from the increase in proteasome activity. And this result you see here. We overexpressed in the wild type cells mitochondrial proteins, and they significantly activated proteasome activity. So this was a great finding, but we wanted to know, is this anyhow beneficial for the yeast cells? And we performed heat stress assay where wild type cells or cells that are deprived of the proteasome assembly chaperones are exposed to heat stress. And we expressed on top mitochondrial proteins that were partially mistargeted. And yes, in the wild type cells, cells where it's overproduced mitochondrial proteins were protected from the heat stress. This protection got lost when the proteasome assembly chaperones were deleted. So that meant that beneficial effects of mistargeted mitochondrial proteins is dependent on the fully assembled proteasome. So this was a first great finding describing a post-transcriptional response of originating from mitochondrial dysfunction. And the study was done in yeast. So now we asked, is this anyhow relevant for a higher eukaryote? And there it comes, the small nematode, C. elegans, that we introduced into our research. I need to show this movie because it's a great model organism. It's small, it's easy to grow in the laboratory. It has a very short life cycle. That's why it's one of the pioneering model organisms in aging research. And we did a similar approach that we did in yeast. We knocked down a mitochondrial import component and first thought to see whether this has any beneficial effects on the worm, and we measured lifespan. So, as I said, have about a 3 weeks life cycle. The animals exist in hermaphrodites. They produce embryos and go through 4 larval stages until the animal reach a reproductive adult stage. From there on, we can follow how the animal ages. And this we can measure by looking at the animal every day, whether it is still moving or not. And we can display this in a graph. And yes, indeed, we found that animals, wild-type animals with compromised import machinery, had an average increased mean and maximum lifespan compared to control animals. So that gave us hope that our findings in yeast could be also true for a higher organism, higher eukaryotic organism. So next we checked whether in our system we also have changes in proteasome activity. We took a little different approach. We took a worm, and the experiments have been performed by Michał Turek. We took a worm that expresses a dendra-2 protein. And when you shine a blue light on this animal, the green fluorescent protein changes its appearance and it appears in red. And then you take some time. After some time, you observe the animal again and count, measure the remaining red fluorescence. And this difference in the red fluorescence is given as the proteasome-dependent degradation. And we express it in a graph. And yes, indeed, to our great findings, we found that in our model with the compromised import machinery, we have also increased proteasome activity, quite similar like in yeast. Now we still wanted to know if lifespan increase upon the compromised import machinery depends actually on the fully functional proteasome. So we turned to a worm that has a deletion of one of the subunits of the proteasome, RPN10. Actually, this is the only deletion that is viable in worms because obviously proteasome subunit proteins are essential and compromising their function has a strong effect on the animal's life. And as in this mutant, we see that we have an accumulation of ubiquitinated protein species. So this is a good indication that proteasome function is actually impaired. And we did the lifespan assay, and we see again that control animals live to a certain time, and animals with compromised import machinery have a lifespan extension. However, if you combine the proteasomal mutant with the import machinery defect, we're losing the beneficial effect. And this was a good indication for us that yes, indeed, the beneficial effect of the compromised import machinery in mitochondria is dependent on the fully functional proteasome. So to sum up this first part, we found that mitochondria that are deficient in mitochondrial protein import machinery accumulate pre-proteins in the cytosol. These pre-proteins lead to the activation of the proteasome, most probably through increased assembly rather than new production of proteasome subunits. And this increased proteasome degradation capacity of the proteasome is beneficial for stress resistance in yeast and helps to extend lifespan in the worm. Now, I think for future work, it will be important to see whether the beneficial responses that are activated here, they also somehow help to feed back to the mitochondria. We don't know this. Also, the question is whether it's possible to define this step a bit more, where the pre-proteins activate the proteasome. So it's not quite clear yet how this on the molecular basis would work. Now, I showed you the first part about the proteasome, but every signaling that exists needs to have a starting point, so a signal that activates it. So what other signals mitochondria can send? So early eukaryotes were still a bit limited in it, but they had metabolism, produced acetate, which leads to protein acetylation, and always the respiration, and the byproduct of respiration is reactive oxygen species. And we also know now that these reactive oxygen species have an effect on the proteins, especially on protein thiol oxidation. In the present day eukaryotes, the possibilities of signals are much more diverse, and you see it here. I don't want to show signaling from all of them, but you see that one of them is evolutionary conserved or older, let's say, and this is the signaling through the release of reactive oxygen species. So why is this so important? Because reactive oxygen species, they modify proteins, and they do this depending on the strength in two different ways. We can have reversible cysteine modifications, and it's now generally accepted that these modifications contribute to cellular signaling. If the stress gets too strong, we have irreversible cysteine modifications. And this can lead to the damage of the proteins themselves, or that at least are not functional anymore. So why was this so interesting for us? It was interesting to pick it up because we found, or we observed, that in our yeast mutants that had impaired mitochondrial import, different ones, we measured an increase in reactive oxygen species. And this was probably due to an imbalance in respiratory chain components. So now, what does oxidative stress do to proteostasis in the cell? And this is actually something that is known for quite some time. It started with papers from Chris Grant's lab, that if you expose cells to exogenous oxidative stress, here by H2O2, you decrease protein translation in the cytosol in a concentration-dependent manner. And of course, we were able to show this as well in yeast. We were able to show this as well in yeast. Same is true for mammalian cells, for example. So would this effect that you get from adding exogenous oxidative stress to the cells, would we have the same effect on the translation machinery when the oxidative stress comes out from the mitochondria? And yes, we do. So what we found, and this is a very nice example of it, that in a mutant, in a yeast mutant, where you have compromised import machinery in the inner membrane, you have first at permissive temperature. The wild type and the mutant have similar translation, global translation. But once you shift it to restrictive temperature and you activate the mutant and the increase in ROS production, we have decrease of protein synthesis in the mutants compared to the wild type cells. So for some reason, the ROS that are produced by the mitochondria also affects cytosolic translation. So what's so special about it, since it's already known, and actually translation attenuation, so the stop in translation, is a very general stress response, and it's induced upon many different stimuli. And you see it when you shift cells to non-favorable growth conditions, viral infections, heat stress, or when you induce any kind of stress in the endoplasmic reticulum, or you have lower nutrients, and you decrease the overall translation. And it makes sense because the cell wants to protect itself from producing even more proteins that it cannot deal at the moment with when the stress levels are too big. But what brought our attention to it is how the translation is actually stopped. So translation can be regulated at the step of initiation, elongation, or termination. And what was already known for some time is that when you add exogenous oxidative stress to the cells, translation is stopped primarily at the initiation stage. And we have a disassembly of polysomes. And this you can see in this profile and the quantification. So upon H2O2 treatment, we have many more monosomes compared to the polysomes. However, when we do such an experiment in our mitochondrial mutant, where we have the translation stop as well, we do not see this shift. So somehow, and polysomes are more maintained, somehow the different signaling mechanism seems to be activated that halts the translation at a different stage rather than initiation. So to find out what could it be, we took an unbiased approach in identifying what kind of proteins could be reversibly oxidized upon oxidative stress. We applied the oxycut method that was originally established by Ursula Jacob's lab, and where you grow cells, you first label reduced cysteines with an ICAT label, and you reduce all cysteines that were previously oxidized and label it with a differently heavy ICAT label. And then you perform the proteomics analysis. We did this in yeast cells, and we did this up on H2O2 stress and in the yeast cells with compromised mitochondrial import. Our study, always there's question whether it's biased towards proteins that are more abundant in the cell, but we couldn't find this, so it was rather equally distributed between less abundant and more abundant proteins. And we found that indeed, upon increase in the oxidative, in reactive oxygen species, we have a small but visible shift in towards the higher oxidation of cysteines. However, it's not a massive shift, and this is to be expected because the cell reacts to this oxidative stress and tries to reverse the oxidation of the thiol groups. But what we found is that, first of all, we found that many more proteins or with thiol groups were susceptible to oxidative stress in the mitochondrial mutant. And this is shown here in dark blue. In light blue is shown the proteins or peptides that we identified to be more oxidized, significantly more oxidized upon H2O2. And the red spots show the overlap between these two data sets. And in the overlap of the data sets, we found that we see components of the translation machinery, including ribosomal proteins. This was quite fascinating because these sites that we identified in the ribosomal proteins, they fall into a cluster and into a site where zinc is bound within the ribosome that is so far thought to be there for structural reasons. So what we identified are components of the translation machinery, including ribosomal proteins that can undergo reversible oxidation upon mitochondrial released reactive oxygen species. And when you try to imagine this, that this could be potentially a new way of regulating translation, which would be non-invasive because it does not require the disassembly of the ribosomes to halt the translation and then de novo synthesis. So to summarize this, what we found is that mitochondria under oxidative stress, they signal through the release of reactive oxygen species to the translation machinery, and presumably oxidize susceptible residues in ribosomal proteins that may cause a halt in the translation machinery. Of course, we have to go a long way still to prove that this mechanistically really works like this. But one can easily think about the benefits of this. So first of all, it decreases the protein load for the mitochondrial import. It's beneficial for cellular protein homeostasis. And there is a third way that one could think about. It's whether these changes in the translation apparatus could maybe lead also to a selective translation. And selective translation means that there are mRNAs that are preferentially translated upon the stress conditions, and those contribute to the recovery of cellular protein homeostasis, or maybe as well to the recovery of mitochondrial protein homeostasis. So the subject of selective translation is ongoing in the field already for some time. But now to combine it with ribosomes that are changed, let's say, from a canonical ribosome to a specialized ribosomes, to make this link, we still have to go a long way. However, there are few indications already that the changes in the ribosomes itself can lead to a different translational output. And I only want to present this kind of summary slide that depicts a few examples of ribosomal proteins that are changed in their stoichiometry, and those have been found to preferentially translate proteins, for example, for vitamin signaling, or important for extracellular matrix or virus amplification. And yes, there are also paralogous ribosomal proteins that are preferentially incorporated in the ribosomes and are necessary for growth on respiratory medium, so for mitochondrial function. Besides ribosomal changes in ribosomal proteins, there could be also ribosome-associated proteins, and there have been also a few already identified that help promote mitochondrial function, or, for example, function in the endoplasmic reticulum. However, as I said, while the concept of special ribosomes forms more and more, also due to the pioneering work of the Barna lab, to really show that this kind of specialized ribosomes also change the translational output, still much more work will be necessary to perform. So to round up my talk today, I hope I could show you that mitochondria are very versatile organelles. They produce a lot of signals, and they induce many different signaling pathways. The first ones identified more than 30 years ago as the transcriptional signaling. But in the last years, so much effort has been put, and many pathways have been identified that are post-transcriptional and even post-translational. The future research will only show how much these pathways are actually linked. And in my opinion, already now we can see some overlap, and maybe also depending on the complexity of the organisms, different signaling pathways will be cross-talking with each other. Now, I want to leave you with this take-home message, which is looking at pathologies, human pathologies linked to mitochondria, we might not even only look at mitochondria itself, but at the surrounding, at the entire cell. And I think we will be hearing much more in the coming years about potential therapeutic targets that can be exploited to combat these mitochondrial linked pathologies. With this, of course, I am coming to the acknowledgements, and I showed you today work that has been done by me and my colleagues in the laboratory of mitochondrial biogenesis, headed by Agnieszka Hacińska. For sure, I forgot people to include here, and many of them are now already alumni in the lab. And great impact, of course, in the question about mitochondria and the proteasome was the team around Lydia Vrubel. And only recently we have been able to complete the complementary work in C. elegans that was in part initiated by Krzysztof Drabikowski in IBB to provide preliminary experiments, and then was carried on by Marysia and Michał. And I also worked together with a great team on the connection between the mitochondrial and the translation. And it was a combined effort between us working on yeast and Łukasz, who did all the mammalian part of the project. Of course, none of the great findings could have been done without the laboratory of Bettina Warscheid at University of Freiburg. And there together, I worked on the OxyCat project with the postdoc Ida Supans. I wasn't able to show you many of the results that contributed to the success of the stories, and that have been performed in collaboration with the laboratory of Andrzej Dziębowski and Marta Koblowska. And more recently, I collaborated with Marek Tchewski, who taught us much more about ribosomes and to handle them, and we are continuing this project. Last but not least, of course, I don't want to miss the opportunity to introduce you to my lab that I'm running now since 2 years. That maybe sound like a long time, but when you start in an empty room, there's actually quite a long way to go to get to the results. But now, mostly during the time of pandemics, we were able to create a great team that's pick up some parts of the translation projects, and Karolina and Ramin are mostly involved in it, and we are looking now much more into what is actually how the ribosome can be modulated upon cellular stress conditions, and finally, what does the ribosome translate under these kind of conditions. And we also have a great project going on led by Somaya and Ismail. They're investigating how actually folding apparatus, folding chaperones adapt to translation attenuation. And we're greatly supported by previously by Marta and Sarah, and now by Monica in the laboratory, who holds the whole team together. And with this, I'm finishing my presentation, and I'm looking forward to the questions.
Great, Ulrike, many thanks for a very interesting presentation. you can stop sharing, right? Sharing the lecture. And now let me check the Q&A form. And let's start from the top or from the bottom. As I told to participants, you can write your question in this Q&A. Let's ask the first question. Do old mitochondria have a protein import problems? If so, it's wrong to try to fix it in anti-aging effort. I do not understand the second part of the question, so let's focus on the first part. Do old mitochondria have a protein import problem?
Mm-hmm. It's a great question. Honestly, I'm not right now recall a specific example that I can give you. Yeah, I think yes, the mitochondrial imports is impaired during the aging process, but we need to find out, I think, why it is impaired, whether this is because of unhealthy protein modifications that destroy the proteins, including the import machinery. I think we don't know this, and it needs some more research to be done on this. What was the follow-up on this?
Okay, the next question is concerning the determination of ROS level. The person is asking you what is a recommendation, what is the best assay to follow ROS? Experiment which we know is very tricky and complicated.
Yes.
So
when we did this initial experiment, we used the ROS sensors, chemical ROS sensors, that you incubate with the alive cells. They are taken up by the cells and are oxidized by themselves, depending on how much ROS you have, and then they are trapped in the cells. And you can have the fluorescence readout for this. It depends on your model. I think right now it is much more fashionable to do genetically encoded ROS sensors. And if I'm going to, but it needs establishment. But I think there are many people working on genetically encoded ROS sensors, and there are many different ones available depending on the compartment of the cell as well. And if I would do this once more again, I would put effort into getting genetically encoded ROS sensors. However, yes, that's the answer to the question.
The next question is concerning your presentation and is following. How would you combine part 1 and part 2 of your talk in one model?
If it concerns part 1, the proteasome, and part 2, the translation, as far as I understood, then this is combined in one model because it happens at the same time. And I think you cannot unlink it, because the production of proteins and the degradation of proteins, it's through the proteostasis network, it is linked. And it's not possible, in my opinion, to change one part with one system without changing the second system. So they rely on each other, and they translation, protein production, and degradation, they depend on each other. So it's just naturally that first, if you have precursor proteins accumulating in the cytosol tries to remove them by degrading them. But at the same time, it also lowers the load of newly produced proteins while decreasing the protein translation. Yes?
Okay. The next question about RNA. Since cellular RNAs, mostly tRNAs, are modified with sulfur, do you consider that observed responses of the ROS translational alternation can originate also from oxidative damage of RNAs?
So yes, this question comes quite often, and once we should really try to address whether we have damage there. However, my answer is always to this. There is oxidative stress causes damage to the RNA, and there's no doubt of it. But in our system, this effect that we see on the translation, on the production of the proteins, it's quite fastly reversible when you remove the stress. So there I see the argument for that we have maybe a minor amount of damage in the system that we are studying in a way that it doesn't affect the system that much that we would need to produce new components of the translation machinery. Therefore, the response is much too fast and it's much too fast reversible. But I agree, proper measuring of damaged RNA would finally answer this question.
Okay, the next question concerning the lifespan, and it is following. Is the effect of proteasome activity on lifespan or stress response dependent on organism age?
You mean that whether the whether the proteasome activation depends on the age of the animal. So the way the experiment is done is that we decrease the or we knock down the import component at an early stage of the animal. So when it's practically just after it hatches from the egg. And then most of the knockdown of the import component is performed throughout the life of the animal. So what has been already shown before is that these beneficial responses connected to mitochondrial stress, they are the most effective when you apply them early in life in the animal. And this is true for other mitochondrial stressors, maybe also for this one. We didn't perform a particular experiment where we would expose the animal at a later stage, or let's say just the adult stage, to the knockdown of the mitochondrial import component. Of course, this could be done to supplement the experiments.
Okay, and because it's 2 o'clock approaching, so the last question, it is following. For the proteins that are targeted to mitochondria and other cellular compartments, can the mitochondrial stress signaling and global changes in protein translation induce redistribution of their peptide pools prior to import into mitochondria?
So this is a great question, and I think there are these kind of pathways now studied, and it's closely relates with the function to the ER. So indeed, it could be that proteins that are pre-proteins that are produced in the cytosol, and they cannot be imported. There are new pathways that show the involvement of the ER. That there are proteins on the ER surface that pick up this kind of mitochondrial proteins that are mistargeted in a way, and they try to target them to the mitochondria, or try to or help in targeting them to the proteasome. But these are the first studies that came out in the last years, and I think it also still needs more research to say how other organelles can be involved in this redistribution.
Excellent, Ulrike. Many thanks for a great talk.